Hemostasis refers to the sequence of events that take place to seal damaged or injured blood vessels to prevent further blood loss. There are 3 overall steps involved. First is the vascular spasm, second is the formation of the platelet plug, and third is called coagulation. The immediate effect of damage or trauma to the blood vessel is what's called vascular spasm. Smooth muscle lining the walls of the blood vessels will contract, and this decreases the diameter of the blood vessel where the injury has occurred, which is going to accomplish a few things. It slows blood flow to the injured area, which slows blood loss, and it promotes the formation of the platelet plug and begins activating the clotting factors, as both of those will occur in areas of slowed blood flow. Platelet plug formation, as its name suggests, occurs when platelets begin to attach themselves to the site of the injury to form a temporary seal. The platelets that initially attach themselves release chemicals that promote and maintain the vascular spasm, in addition to recruiting additional platelets to the site. As more platelets are attracted to the injury, they continue releasing more chemicals in order to recruit more platelets, and the platelet plug begins to grow large enough to seal the wound. This is one primary example of a positive feedback mechanism. The final step in hemostasis is called coagulation, which is the formal name for the formation of a blood clot. In general, coagulation ends with the activation of fibrinogen to form fibrin, which is a protein mesh that will stabilize the platelet plug in addition to nearby erythrocytes or red blood cells. Coagulation works like a cascade, with 13 clotting factors that are activated in sequence to create the blood clot. As discussed earlier, most of these clotting factors are produced by the liver. So if a person has deficiencies in a few of the clotting factors in the cascade, that results in a condition called hemophilia, where a person will bleed excessively due to blood clots not being correctly formed. Overall, there are 2 pathways that lead to the creation of the fibrin mesh: the intrinsic pathway and the extrinsic pathway. The intrinsic pathway to coagulation is activated by clotting factors within the bloodstream and occurs when the clotting factors come into contact with foreign materials. So that is, materials that are not the tissues and the cells that line the inner walls of the blood vessels. One example of a foreign material is glass, which explains why blood will clot when placed into glass sample tubes. If that glass tube happens to be coated with heparin on its inner surface, the heparin would prevent the activation of the clotting factors. The extrinsic pathway to coagulation occurs when blood plasma comes into contact with damaged tissues that are external to the blood vessels, as the term extrinsic means external to. So both of the intrinsic and extrinsic pathways lead to a common pathway of coagulation, which is where fibrinogen is activated into fibrin and becomes the fibrin mesh that helps to stabilize the platelet plug.